Today we're going to continue our discussion of Chapter 9, Section 1, and also talk about Chapter 9, Section 2. And this is more of the tools of biotechnology. We're starting out here with a picture of a process. This is a multi-step process that is our next tool, and the tool itself is just referred to as gel electrophoresis, which is just a big fancy word that means we are using electricity to separate DNA fragments by size. So what we have here is a power supply. If we're using electricity, we need a power supply. We have a cathode and an anode. That's your negative and positive terminals. There is going to be a gel. This rectangle is a gel made out of something called agarose. We'll talk about that on the next slide. And then it has these indentations or wells where you can load DNA samples. You put the DNA in there. It's floating in a buffer. A buffer is something that can conduct the electricity. And then you turn on your power supply. The electricity will pull those DNA fragments through the gel and cause them to separate by size. So you can see the basic steps are listed here to the left of the diagram. First of all, notice that we have DNA in fragments. We've already talked about how to cut DNA, and we would use restriction enzymes. We mix the restriction enzymes with the DNA and it chops it up into fragments. We talked about the electrical current. The DNA moves from the negative side to the positive side. So if you know anything about electrical charges, that means DNA is moving towards the positive. Opposites attract. That means DNA has a negative charge, and that causes it to be attracted to the positive side. If you think about the functional groups in a DNA molecule, that nucleotide has a sugar, it has a nitrogen base, and it has that third part, the phosphate group. Phosphate groups have a -2 charge, so DNA molecules are negatively charged. Now, as that DNA moves through the gel, the small fragments can move much faster than the large ones. So on this end of the gel, closer to the positive side, these pieces of DNA are small. The large pieces of DNA were stuck back here closer to the beginning. And so then we have all of these different lines that represent DNA fragments of various lengths, from small on the right to the large on the left. And we would call each line a band of DNA on the gel. So let's talk about that gel and what it's made out of. It is an agarose gel, and if you remember what OSE stands for, you know that's a carbohydrate. So this is a type of carbohydrate extracted from seaweed. This picture here is taken with an electron microscope, and you can see inside of the gel there are all these holes that DNA can move through. But it's kind of thick. It's like if you were trying to walk through the woods and there was a lot of underbrush that you had to try to make your way through. The gel itself is going to look like this. So you can't see those holes to the naked eye. You have to use a microscope to see it. But here are all of those wells. That's where you would load your DNA sample. This gel actually has 2 rows of wells, so 2 places where DNA can be loaded. That's the kind of gel we will use in class. And as the DNA is pulled through the gel towards the positive side, it has to make its way through all of these openings. So if we use that forest example, you as a full-grown human would have a much more difficult time traveling through really thick underbrush compared to something like a chipmunk. A chipmunk would be able to just run right through there, through those openings with no problem. So that's why the small pieces of DNA are able to move farther through the gel than those large pieces of DNA. Now, what we see at the end of the process looks like this. You'll notice this is blue, but this original gel was not. That's because we have to stain the gel. We can't see the DNA. The DNA is clear, so we have to use a stain that sticks to DNA, and that shows us the bands of DNA. So all of these dark blue lines are DNA that have been stained, and again, This is the top of the gel. That's where the DNA started. So the larger bands are going to be closer to the top. Smaller bands are going to be closer to the bottom. So this would be the largest fragment size, and this would be the smallest fragment size. You might notice on some gels there are really dark, thick bands versus really faint bands. That just has to do with how much DNA is in there. So this would be a faint band. That means there's not very much DNA of this fragment size, but this really thick band in the middle, that means there's a lot of DNA of this fragment size, whatever that size is. Our next tool is called polymerase chain reaction. This is just called PCR for short. You can just call it PCR. It's a lot easier to write that out than polymerase chain reaction. The purpose of PCR is to copy DNA. And this is a way that scientists have figured out to replicate the real-life process of DNA replication in a test tube, in a machine, in the laboratory setting. And this has revolutionized the way that we work with DNA, because now it is very easy to relatively quickly make a lot of copies of a certain DNA sequence. So this is really helpful in forensics. For example, if you were to collect a drop of blood from a crime scene, there's not a whole lot of DNA in the blood, not enough to really see it on the band like what you see here. So what you do is you take that DNA from the blood and you use PCR to make millions or billions of copies of it. So there's enough of it there for you to be able to see it with the naked eye. So this is a really important process that happens before a lot of other biotechnology or forensics processes. You have to do this before gel electrophoresis. Otherwise, you don't have enough DNA in your sample to actually see it. The steps of PCR, there's just 3 major steps, and these 3 steps cycle over and over and over again. Our first step is called denaturation. Well, you already know what denature means. This is when we are using heat. The machine for PCR, this is one example of a machine here. You put all the tubes here on this heating block, and that heat will heat up that DNA so that it starts to separate the strands. So this is mimicking the action of helicase, but we're not actually using an enzyme for it. We just use heat. It's a lot easier in a lab just to heat it up and make it denature rather than add a very specific enzyme. Our second step is called annealing. Annealing is sticking to something. So the thing that sticks to it is primers. You will remember when we learned DNA replication, you know that there has to be a primer at the beginning of the segment of DNA you're copying. So if we're trying to copy a gene in the process of PCR, the scientist has to add primers to their sample so that that copying process can get started. And the third step is called extension. This is when a DNA polymerase is going to start at the primer, and then it's going to create this new strand of DNA. It creates one on this side and it creates one on this side. And that works the exact same way as what we learned when we learned DNA replication. The polymerase adds complementary nucleotides always on the 3' end, and it builds that new strand of DNA. All 3 of those stages together is known as a cycle. And PCR is going to cycle sometimes between 20 or 30, maybe even 35 times. And if you can think about each time that cycle happens, you are doubling the amount of DNA. So if we have this DNA, we go through one cycle, one piece of DNA is now 2 copies. Well, we go through a second cycle. Each one of those pieces gets copied again. Now we have 4 copies. Go through cycle 3. Now we have 8 copies, and so on and so forth. If we go all the way out to 30 cycles, we can have 1 billion copies of this DNA sequence, and that is plenty of DNA for us to do whatever forensics test we need to do. So that's going to be it for our copying DNA and 9.2 notes. We are going to spend a little bit more time with this in class. You're going to actually do some of these processes, make your own gel, and see how all of this works. So if you need to revisit this video at any time, please do so to understand the underlying biology of the process. And if you have any questions, as usual, please make sure that you ask those in class.